Hello everyone, welcome back to another video. This is my fifth time recording this and I'm a bit annoyed now because I've been making silly mistakes when I'm making it. So forgive me if I make mistakes this time. So today we'll be looking at homeostasis. This is the part one and I will try and upload part two tomorrow. This is IGCSE Biology 0606. Let's get started. Homeostasis. Homeostasis is the maintenance of a constant internal environment. Now, when you talk about homeostasis, right? Homeostasis. We have three kinds. The first one is thermoregulation. Thermoregulation. And now, as soon as you see thermo, I'm sure you know that it has to relate with thermal or temperature. Then we have glucoregulation. Glucoregulation, and this has to do with your blood glucose concentration because gluco means from glucose, right? Then we have osmo regulation, which is your water level. Glucoregulation, yeah, right. But luckily for IGCSE, you only need to know thermoregulation and glucoregulation. I'll do thermoregulation in part two of this video, and this is what we'll do today. Okay. Homeostasis looks at controlling internal conditions. Internal conditions. And you know that body temperature, body glucose, blood glucose concentration, and water levels in our body are all internal conditions, not external, right? Such as temperature, yeah, we said it. That homeostasis helps cells to work as efficiently as possible. They keep a constant temperature. That means they are regulating the temperature, which has to do with thermoregulation of around 37 degrees Celsius, helping your enzyme to work at an optimum rate. Keeping a constant amount of water. Again, osmoregulation, because it has to do with the water and a constant amount of it. Means that your cells are not damaged by absorbing or losing too much water by osmosis. Yeah, that's why it's called osmoregulation because it comes from osmosis, right? Yeah, you may have wondered why is it not hydroregulation or something like that. Yeah, that's why it's osmoregulation due to it being controlled or due to the water being absorbed or lost by osmosis. Keeping a constant concentration of glucose. Constant concentration of glucose, that's where glucoregulation comes in, means that there's always enough fuel for respiration. And we all know that respiration is basically glucose plus oxygen giving us water and carbon dioxide and energy. So for this energy to be released, we need our glucose. That's why the constant concentration must be maintained. Then there's the first thing, glucoregulation, that we'll look at. In glucoregulation, we have two main organs that help in controlling the BGC, blood glucose concentration. The first one is our pancreas, and the second one is our liver. The blood glucose levels are monitored and controlled by cells in the pancreas called islets. So these islet cells are scattered all across the pancreas that detect the rise and fall in blood glucose concentration or BGC. Okay, don't go and write BGC in your exams. I'm just saying it so that I don't have to repeat blood glucose concentration 1,000 times. The pancreas produce and release different hormones. Now, for glucose regulation, we are releasing what hormones? And they said so basically, yeah, we have two hormones that can be released depending on if the BGC is high or low hormones. So these two hormones are insulin and glucagon. Insulin yeah, is when your blood glucose concentration is very high. And glucagon is released when your blood glucose concentration is low, and we'll look at how and why. The hormones help the liver to control the amount of glucose in the blood. Changes in the glucose concentration. Let's say I eat a lot of carbohydrates and they eventually break down to glucose. I have high glucose concentration now. What is going to happen? What is going to happen is that the islets will first detect the change in the pancreas and secrete insulin into the blood. Now, great, I have insulin into the blood. And how does insulin travel to the blood? It dissolves in the blood plasma, right? Make sure you know that. When insulin reaches the liver, okay, so let's say we have pancreas here and they're connected like this using vessels to the liver. So pancreas have secreted insulin and it has flown with the blood plasma to the liver. Now what is going to happen is that it will cause the liver to absorb glucose from the blood. Now the blood doesn't just have this hormone insulin, it has a lot of other things, right? So the blood glucose in it will be absorbed by the liver because I have an excess of something. I have too much of it. So how will I decrease the too much so that I bring it back to normal concentration? I will absorb some of it or store it. Yes. The glucose will be stored by converting it to glycogen. Okay. Make sure you don't confuse yourself with glycogen and glucagon. Okay, and stored in the liver. Some is used for respiration, and this will decrease blood glucose concentration to normalize it. Okay, but how will that happen? What will happen is that you see glucose. Glucose is soluble and it easily dissolves in blood plasma. But glycogen isn't. Glycogen is insoluble and that's why glucose gets converted to glycogen and it doesn't flow with the blood. Okay, and that's why it's stored in the liver. If you don't understand anything, feel free to comment down or if, in fact, email me and I'll try and better explain it, just so you know. Then we have low glucose concentration. Maybe it's been hours since I've eaten something and my blood glucose concentration has gone really down. Again, what will happen? The islets in the pancreas will detect the change and secrete glucagon this time. It's important to remember the names insulin and glucagon. I mean, you get it with practice, so don't stress yourself too much now. When glucagon reaches the liver, now you know what is happening is that you have very less of something, and you'll have to increase it so that it becomes normal, right? So this glucagon will go to the liver and cause the liver cell to break down the glycogen into glucose and release it into the blood, which will increase blood glucose concentration. Now you have this glycogen, right? It's because it's been stored. Remember when we talked about high glucose concentration, it went and stored the glucose as glycogen. Now that you have low glucose concentration, this very glycogen is going to be converted into glucose so that you get normal glucose concentration. Okay. Then we have negative feedback and set points. Um, in practice, our BGC does not stay absolutely constant. A normal concentration is somewhere between 0.8 and 1.1 milligram per cm cube, mg per cm cube of blood, and this range of normal values is called set point. Okay, let me explain that. So what it's saying is that in your blood, right, the blood glucose concentration is not going to be a fixed value. Fixed value? No, it's not going to be a fixed value. It will be a range. It's not going to be just, okay, I have 1 milligram per centimeter of cube or centimeter cube of blood. Oh God, glucose concentration in the blood flowing in everybody. It varies, and the normal values that it should be within is 0.8 and 1.1. And these values are called set points, essentially. Yeah. So set point is a normal value or a range of values. Now we say normal value because when you look at thermoregulation, body needs 37 degrees Celsius, and over here it's a range. That's why you say value or range of value for a particular parameter. Now negative feedback, it's a mechanism that detects a move away from the set points and brings about an action to take the values back towards the set point. Now read it carefully, pause the video and read it carefully. It's saying that it's a mechanism that will first detect the change. I'm not necessarily saying change in your glucose concentration. I've said change, detect a change away from the set points. Now, this is because we are generalizing it for thermoregulation, glucoregulation, and osmoregulation. So once it has moved away from, okay, let's say I have my set points here. Okay, and any value over here is fine. But as soon as the value goes outside the set point over here, or maybe outside the set point, what will happen is I'll go through mechanisms called negative feedback and I'll do some action or the other to bring back this within the set points for here or here. So in negative feedback, we have set point. There's a measuring device that tracks whether the level is within the range of set point. If the level goes outside, then triggers events. These events are actions to happen so that the level brings back into line again. Then we have diabetes. Diabetes, we have type 1 and we have type 2, but we'll be looking at type 1 only. This is caused when the cells that secrete insulin die. It's not certain what causes diabetes, but it's thought to result from the body's own immune system attacking and destroying the cells in the pancreas that secrete insulin. This type of diabetes usually develops when a person is young, or in fact, even genetics, right? Yeah, so like if your grandparent or your grandmother had it, your dad probably had it, then you're also likely to have it. Normally, the person triggered the secretion of insulin from the pancreas when BGC is high, but in type 1 diabetes, this would not happen. The blood glucose concentration goes up above the set point and it remains there. Okay, and it remains there. Now we know that too much of something or too less of something is never good, right? On the other hand, we not eating carbohydrates for a long time will cause the blood glucose concentration to drop low. Now you know that you don't have your cells do not secrete insulin. That means you cannot store the excess carbohydrate, and that is why you do not have any glycogen that can be converted to glucose. You don't have any glycogen that can be converted to glucose when you have low blood glucose concentration. And when this happens, it can cause you to feel tired because you have low blood glucose concentration and you may feel confused and you have an irrational behavior. So hyperglycemia means high blood glucose concentration. Hypo means low blood glucose concentration. So these are some symptoms of diabetes type 1. In type 1, we said that we have two types, hypo and hyper. So in hyperglycemia, you feel unwell, you have a dry mouth, blurred vision, you feel thirsty, and your heart rate and breathing rate will increase. In hypoglycemia, you feel tired, show confusion, and irrational behavior. Yeah, so you basically do have to memorize this, or you have to memorize. There's no trick to some things, right? Then treatment for diabetes. Eating little or avoiding large amount of carbohydrate, because you know that it's carbohydrate that eventually breaks down into glucose. So if you maintain a particular amount of glucose or carbohydrate that you intake, you're likely to maintain a constant concentration of glucose in your blood. We have injecting insulin. Because the cells that produce insulin are no more there, if even if you take in lots of carbohydrates and you inject insulin in your body, that will help bring down or normalize blood glucose concentration. We have two types of insulin, rapid action and long action. Rapid action is when you take um is the one that you take just before a meal or after a meal. After the meal is depending on how much carbohydrate you took or you ate, and before is you are going to judge or predict how much you take, and then that is how much of insulin you will take. So that's before or after your meal. And long acting, sorry, is when you take the insulin in the morning, let's say. at the same time each day, so that a constant production of, so that insulin is in your body, which will help normalize your blood glucose concentration when you take excess glucose, right? So this provides a background dose of insulin. And that is that. Pretty informative video. Thank you for watching. Make sure you like, share, subscribe. And make sure to stay tuned for the next video, which will probably come out tomorrow. I'll film it. It's what? It's now 2:25 a.m. here, but I'll try and film it and post it. Thank you for watching once again. See you in the next video.